And I should say, Dave, thanks very much for all the support over the years. My head of department's here, Alyssa, so thank you more recently for all the support. So I'm going to talk a little bit about our research. We do look at molecules, but the binding kind of concept in our research is the way in which they interact, and so I'll talk through this. This funny looking picture that you see here is actually a diffraction pattern, and this was the very first diffraction pattern that I was able to get as a PhD student in Sweden, and actually that can be translated into a protein structure. I still can't really tell you how you do that. There's maths and models that are involved in that, and the computer did it. But that really captured me. I mean, these diffraction patterns are absolutely beautiful, and then the structures that came after that were also beautiful. So this is Science is People and Ideas and Discussions and Connections. And so this was my first, as a researcher, having my own group at University of Canterbury. I'd been there for about 18 months, and this was our first Christmas party. And there's a few people to acknowledge in here. Firstly, it's Juliet Gerard sitting down there, and she was my mentor as a PhD student, and she really got me going in science. She got me to Sweden to get that picture, actually. But also this was my first group, and I really want to just emphasize how well they've done, because I'm really proud of the success they've had. And so Kat Donovan there was my first PhD student. She's now a scientist at Harvard, and if you ever look it up, she's got probably the best track record for a researcher from New Zealand in the molecular sciences. She's absolutely amazing. Muge is a professor now in Germany. Jeremy's an academic at Warwick. Rachel just started her job as an academic at the University of Sydney. And then Amy over here is a new academic at the University of Canterbury, so she's one of my colleagues. And in the theme of interactions, she married Eric, and they have had their first baby. And so that's really, they are the ones that started all of what I'm going to talk about today. This is the current group. They're young and ambitious. They call themselves the Young Guns, and I like that. And this year we had a game of cricket. And I won't point them out, but there's the folk in there have done all the work that I'll talk about. So what's the core of what we do? So it's really about the interactions of molecules. And so I spent a lot of time early in my career solving the structure of this, or solving the structure of that, which was fun from an academic perspective. But actually it's the emergent property of how these things come together to do something that is what life is, or the way I define it. Now this, if you can believe it, is actually a painting made by a structural biologist probably about 10 years ago of an E. coli cell. And so you've got a flagellum, and you've got some other stuff, I don't know, things in there. But what he's done is he's painted all the structures of the inside of a cell as best we knew all of those components. We don't know all of them, but most of them. If you focus just at the top there, you'll see that, well, I'll point it out, there's actually two membranes, and we'll get back to that separate the outside of the cell from the inside of the cell. Then on the inside, what you've got is this kind of chaotic mess of molecules that are interacting and doing things. There's lots of interactions going on here. You can't quite see, it's off the page, but there's DNA down here, RNA here, there's proteins making new proteins, all sorts of things going on. So that's really the context. So I'm interested in understanding those interactions, and I'll give three kind of examples of how we try and understand those interactions and why. So the first one is, how do cells import nutrients? And so one of the founding features of a cell or life is that it's separated from its aqueous environment. And the way cells do that is they have this membrane that sits in it. It's a fatty membrane, so it doesn't mix with water. But that's a problem for a cell, because a cell needs to interact with its environment. It needs to bring things in, it needs to push things out, it needs to sense what's out there. So how does it do that? It does that by including a protein. A protein is a polymer that's made by the cell. It's what we eat in our milk and what have you. And so the cell makes this protein, inserts it in, and then it can interact with the cell across that membrane. And so there's lots of examples here of different types of machines that are in those membranes. This is a bacterial cell. It turns out, why would we want to even know about this? Well, most drugs that we use and take interact with proteins that are inserted into membranes. About 60% is the rough gauge. So that's an area, and it makes sense, right? You have a drug, it binds at a surface, it changes the biology. How do we do it? This is a bit of a plug for New Zealand getting together to buy a cryo-EM, Alyssa, only $30 million if the department wants to get one. So this is literally a microscope that you can use to look at molecules at the molecular level. This is a big change in the way in which we've been able to do structural biology. What we end up doing is getting lots or millions of these little pictures, and we average them together, more models, thank you very much, Michael. And in the end, you get these structures that come out here. And you can see actually, not only is it a set structure as that painting would depict, but actually these molecules are moving. They have a lot of motion, and that motion is important. I'll just say down here, Rachel, James, and Michael did all this work. And these were the first cryo-EM structures from the University of Canterbury, actually. Now, it's not just that, and I want to bring this in. This is a MD simulation. So we do, oh, that movie didn't work in the end. There it is. So this is an MD simulation. Jane Allison did this. And of course, those membranes aren't just by themselves. They're in a membrane interacting with fat and water on the inside and the outside. And that really influences how they move. And what Jane was able to tell us was how this particular protein was able to elevate up and elevate down and move molecules from one side to the other. And so we couldn't do that without her. The second one I want to talk about is in diagnostic devices, and that's a kind of a new area that we have with engineering, which has been super fun. And probably many of you have seen the COVID test or a pregnancy test. At the heart of these devices are the interactions between several biomolecules that come together that give us a result. I don't need to go through a lot of detail, but if the sample has the analyte, if you're pregnant, for example, and you're going looking for a particular analyte, there'll be an interaction between an antibody and that analyte. It'll get stuck onto a surface, and you can read that out as a color change. What we've been going with this is trying to develop new chips, plastic chips that are cheap and easy to make, that don't have any electronics and they don't have any pumps, so they're totally no mechanics at all, just a single chip. And the way they work is through capillary microfluidics. So it's capillary force, just like a plant is moving solution up through against gravity to their leaves. So here we've got some dye, the celery is moving that water up to its leaves against gravity. So it's capillary force. And actually the innovation we had here, this was super cool. One of the problems with capillary action is that it's kind of set. It can only really go in one way. You can't stop things, restart things, time things, measure things. And you need all those if you want to develop a diagnostic device. And so what Daniel did, and Claude and Julian, they're down here, what they did is they developed a way of taking a capillary device and stop a flow. They can now restart a flow. They can time things. That means you can do an enzyme assay. You can do way more complicated type diagnostics than I showed you before. And Daniel's taken that, and for the wine industry, gave him some money. I don't know if that's already going. This is a movie, and it's hard to see. This actually does work. It's going a little bit pink. That's because it's picking up the glucose in a juice, and the pinker it gets, the more glucose is there. So this is an actual working prototype. And this is Daniel, of course, having to do some research in France, which was critical. All right, the last one I want to talk about is in food systems. Actually, one of the nice things about being a biochemist in New Zealand is you have to be a little bit you've got to work in lots of different disciplines. And so food science wasn't one I was thinking I would work on, but people knocked on my door and said, Dobbo, would you be interested in looking at this particular thing? Oh, okay. There's $100,000 in it for you. Oh, okay. I'll definitely do it then. So we got interested in food systems, and there are interactions here. Now, believe it or not, the father of cellular agriculture is Winston Churchill, and he said this back in the 1930s, right? We will escape the absurdity of growing a whole chicken by growing these separate parts under a suitable medium. And we're just getting to the point where we can do that. And we're borrowing a lot of technology from medical fields. And so what we can do now, this is what we've been doing. We can take various animals. We've been working with cows mostly. You can take a biopsy. These animals are already dead. This is at the abattoir. And you can extract from that biopsy the different types of satellite cells that would go on to grow muscle, for example. You can take that and you can grow these in media, and then ideally in a fermenter or some scalable way so that you can do it cheaply, and then you can make your steak. That's where we want to get to, a structured food. At the moment, we're at a dumpling. So we've made a dumpling with our folk and collaborators in Singapore. Apparently it's tasty. You can eat it. We can't eat it here, but they did. And so why do we want to do this? Well, for Singapore, I mean, Singapore really drove this for us. And they said, you know, we have a lot of food security issues. We don't have a lot of land, and we really rely on our neighbours. And they had some scenarios where they couldn't import beef or pork all of a sudden because there was a disease. Chicken was suddenly unavailable. This was a real scare for them. And so by being able to scale and grow meat, they do have a lot of technology in a factory, then they can get that nutritious need. Now, in New Zealand, so I went to a symposium on Wednesday and learnt about all the cellular agriculture that's happening in New Zealand. And there's a lot of cool stuff going on. There are folk from Plant and Food that are looking at cells from fish. That seems to be quite a good way to do it. And believe it or not, there are folk that are trying to grow fruit and vegetables in this exact same way. All right? Where does protein interactions come in all of this? Well, it turns out that these cells are super expensive to make. The media is quite expensive. The very first burger that was made this way was something like 300,000 American dollars. Right? That's just for the patty. I don't know what they charge for the bun. And so often these foods are augmented with other types of proteins. And what turns out to be important is the interaction between the cells and the plant protein. So what we've been doing is trying to develop different types of seed proteins. In this case, it's pumpkin. We've got others as well. And here, Rachel Benny made this on our fancy new confocal microscope. Each of these little blue dots is a nucleus. When a muscle becomes a microtubule, each cell has many different types, many different nuclei. So we know that it's turned into a microtubule. And this stain here in red is for the myosin, which is the chain. So it's making the proteins that are necessary for a muscle. And this is happening on the surface of these pumpkin proteins. And that seemed to be really critical. Now, it's not all good news if you're into cellular agriculture. There are lots of ethical issues. It means we can probably sample cells from lots of different organisms. Some of those, like kiwi, probably we don't want to do that here in New Zealand, but people are thinking about what other types of foods could we make in this way. All right, so that just gives you, I guess, a spread of the work we've been doing. Many people to acknowledge, I've already acknowledged the students, but it's a truly international connections that I've had for all of the different projects, and I really thank them and, of course, the funding. Thank you.
Well, thank you very much for that. I'm just thinking about the last bit. So $300,000 for an artificial patty. So can you send those people out to my farm and I'll sell them one cattle beast for $100,000 and it'll have a lot more meat on it.
I'm sure they'd pay for it.
Anyhow, do you have any questions for Ren? Very interesting talk, Ren. Thank you.
Thank you for the very interesting talk. In terms of cellular agriculture, how do you feel that regulation is keeping up with the science in this area?
Well, that's a poignant question. Well, you know, the regulation is changing. I'm of the view that we should have some deregulation of the constraints that we have in terms of genetic modification. I think there should still be oversight for sure, but I think for the most part, what the government's doing at the moment, I think is a positive step for these industries at least.